Hello and welcome, dear friends, to our ongoing session of exploring protein synthesis at various levels. I recommend you to go through my previous lectures of lac operon and tryptophan operon for understanding today's concept of regulation of gene expression at two different levels. Today I will be dealing with one level out of which that is the transcriptional level control. It can also be controlled at the translational level. So we have been seeing protein synthesis. We did see the gene expression with respect to the genetic code, its characteristic features, then transcription, translation. We saw catabolic operon in the form of lac operon. We saw anabolic operon in the form of tryptophan operon. So I hope you have understood all these points, which is necessary for you to understand the point which I am going to explain today. So it is an interesting concept that a cell to avoid wasteful synthesis, that is, a cell will never undergo a wasteful synthesis. I am specially talking about a prokaryotic cell. It will try to avoid a wasteful synthesis, and for that it has a mechanism known as gene regulation. Now it is a very point of common sense to understand that if you have to control the production of a protein, you have to control it before even the mRNA is formed. That will be most efficient. You can also think about its control once the mRNA is formed, then not allowing the translation to occur on that mRNA. That system also exists. We will see that in different lecture. But a prokaryotic cell, the primitive cell which has existed on this earth for millions of years with a special technique of remaining primitive and its some mechanisms of its own, one of the interesting aspect is how it regulates its protein synthesis at the level of transcription. So let's explore that thing. We have seen this at length, how DNA is regulated at the level of the regulatory genes and how the structural genes are controlled by the product of regulatory genes. So the promoter, then the coding sequences, all these things are clear for you. So the gene product, that is protein, to get produced, it needs transcription and translation, and inducible genes are expressed only when they are induced by a inducer. Like for example in lactose was the inducer which was needed to remove the repressor. The whole point of discussion here is the fact that how does a cell really know when to start a protein synthesis, second, how much amount of protein is to be produced, and when to stop a protein synthesis. This is all about the regulation of gene expression. This is what a regulation is. Now if you see the steps, the first step is binding of a RNA polymerase onto the promoter site. That is the first step of protein synthesis. Now if a cell decides whether it needs proteins or not, the best part would be to not allow the RNA polymerase to bind on the promoter. That is the core concept of regulating the transcription. But the things are not as simple as they seem, because let us just compare this whole process in a multicellular system. For example, just for the sake of fantasy, compare it with humans. It cannot be compared, try to understand, but let's compare it with humans. Now for a catabolic type of system, we can decide with our hands and brain whether to eat or not. Cell doesn't have that type of regulatory mechanisms which we function partly with our brain. So the brain of a prokaryotic unicellular system is the enzyme system itself. Not only the enzyme, but controlling the quantity and the time of enzyme production. That is the point in discussion today. So regulating the amount of enzyme, regulating the type of enzyme is what makes the cell energy efficient if it has to survive for long. How does the cell carry out this? So regulation of a gene expression is usually done at various levels. Regulation can be done at the transcriptional level, that is called as the transcriptional control. Regulation can be done at the translational level, that is called as the translational control. And even after the expression, in most of the eukaryotic systems this is seen, post-translational control, that is change of the protein activity by chemical modifications. And transcriptional control is changing the mRNA lifespan. Okay, changing the frequency of transcription initiation. When has the mRNA to be initiated and when not in production. That is all about the regulation at transcriptional level. When we speak about regulation at the translational level, whether the ribosome will be allowed to bind or not, that can be the point in question. So we'll see that in a different lecture. In today's lecture, we are going to explore the transcriptional control of prokaryotic system. There is a marked difference in the regulation of gene expression in eukaryotes and in prokaryotes. There may be common activities or common procedures or common practices in the eukaryotic cell and the prokaryotic cell, but apart from the common things, there is a marked difference. They are totally diverse in their way of regulating the gene expression in their own ways. So we will be mostly discussing about the regulation of gene expression in prokaryotes. Now this regulation of gene expression, as I said, is at three different levels. We will be focusing mainly on the regulation, that is turning the protein synthesis on and protein synthesis off at the level of transcription. So a gene, when it is expressed, you can see there are certain regulatory proteins which bind to the DNA, like the repressor. We saw a repressor which binds onto the DNA, and that actually controls whether the RNA polymerase can further function or not. Okay, one point. Second point is, even if a mRNA chain is initiated, there are certain internal folding sequences in the mRNA itself, which we call it as, there are three names, remember. One is the hairpin and loop structure, hairpin structure. Second is the stem and loop structure. Third is attenuator sequences. These all three can be used interchangingly, means for the same purpose. Similar, this may sound the same when it comes to the activity. They are similar in nature. So attenuation is termination of transcription at a very early stage due to the internal foldings of mRNA. So this is these mechanisms and some more mechanisms we will see. 3 to 4 types of regulation at the transcription level exists in the prokaryotes. Secondly, there is translational level, which I will discuss in my next lecture, so I will skip that part. You just focus on this box here, that is regulation of gene expression at the level of transcription. So you know the concept of operons. I will not repeat it again, but just to give you a recap, operon is a group of genes that are transcribed at the same time. We call it as a polycistronic mRNA molecule, and these are found only in prokaryotes. Bacteria, they are always in a dynamic environment, a typically changing environment in which the nutrient availability increases and decreases rapidly. In one instance of time it is available, in the second instance of time it is not available. So bacteria has to respond to such variations in their environment by regulating the gene expression, by altering the gene expression pattern, that is to switch it on when needed and switch it off when not needed. So it would be a wasteful synthesis if, for example, a lactose metabolizing enzymes are synthesized when there is no lactose, isn't it? But lactose is the only available source of carbon in bacteria which induces the lactose metabolism. It has to induce, when lactose comes in the environment, it has to induce its own intake and degradation. If not, it will die. Okay, so there is a sort of genetic regulation in the bacteria at the level of transcription, which is very energy efficient. Means if there is a question which is the energy efficient mechanism, regulation at the transcriptional level or translational level, obviously the answer is at the transcriptional level. Regulating at the transcriptional level will make it more energy efficient as compared to the translational level. Now what actually happens at the transcriptional level, how the on and off mechanism functions, we have seen this in the lac operon, we have seen this in the tryptophan operon with respect to certain repressors which are formed. Okay, so I'll just give you a bird's eye view of the process which is common for most of the operons. When we study lac operon and tryptophan operon, remember these are models, examples which represent the other operons. So there is a regulatory gene, there is a structural gene, and the mRNA which is formed, it is regulated, it is, that is the attachment of the RNA polymerase on the promoter is regulated by a repressor. This is a protein product, and this repressor usually is always 24/7 bound to the operator side. Okay, it is always bound because, so see, in the cell there is a constant urge by the metabolic system to produce. It will not stop, but there is also a constant production of repressor which will always keep binding at the site of operators. So there is an automatic control, and if there is an induction, that is the first part or the first type of control of gene expression at the transcription level is by regulating it by repressors and inducers. The example, best example is lac operon. So this, as I said, the repressor is always bound at the promoter, always it bound, and the operator is always bound at the promoter, that is RNA polymerase. Sorry, listen once again. The RNA polymerase is always bound to the promoter and the repressor is always bound to the operator. Means they are in a Lockhorn situation. If the repressor is removed, the time it is removed, RNA polymerase will rush into the structural genes. Now for this to happen, repressor has to be removed. Who removes the repressor? The inducer removes the repressor. Inducer, what is the inducer? The substrate for which these genes are to be transcribed. Lactose is a mere example. It can be any other substrate, it can be any other pathway. For that particular metabolism, when the enzymes are needed, think about the metabolic map which you have studied in your metabolism. Those thousands and hundreds and thousands of enzymes which are needed to catalyze each and every metabolic reaction, to carry forward one metabolic reaction to the next and next and next, and ultimately derive energy. Each step is needing an enzyme, and each step is a protein, and each step has one gene for that. One gene, one polypeptide, one gene, one enzyme is the concept which Bidley and Totem, these scientists, gave. Earlier it used to be called as one gene, one enzyme hypothesis, but one enzyme would be a bit more too specific, so nowadays we call it as one gene, one polypeptide, which could be an enzyme, of course. So this enzyme, which is a protein, is having a gene not only for lac operon or not only for tryptophan operon, and all these genes on the E. coli chromosome, or for that fact chromosome or DNA of any bacteria, all the genes, they are having thousands of molecules of RNA polymerase which will be always bound to the site of promoter. There will be an adjacent operator gene in all the operons which will always be bound by a repressor, and that repressor will be specific for the specific inducer. It could be lactose, it could be arabinose, it could be maltose, it could be anything. And that inducer, when it comes, it inactivates the repressor, it removes the repressor, and the gene starts to get expressed. So regulation by repressors and inducers, that is example which you know is lactose. Lactose, when it attaches to this repressor, it inactivates the repressor, and an inactive repressor is removed from the site of the operator, and this A, B, C, D, whatever the genes are there, they are transcribed and the enzymes are synthesized. So once this substrate is over or finished, there is no substrate, there is no lactose, and suddenly the repressor becomes active and again binds to the site of the operator. Okay, so operator, promoter, these are needed, always needed side by side for proper regulation. And repressor is a protein which is produced by, in lac operon it is produced by I gene. Now remember that this I gene may be near to promoter and operator or may not be near. It is not near, it can be present anywhere in the gene cluster. It just needs to synthesize a repressor and leave it in the cytoplasm. From the cytoplasm, enzyme substrate specificity, it will always be very specifically attached to the operator site. Repressor will always bind to the operator site. This is the picture 24/7 in the cell, a constant picture. Always this thing happens. Now, this is how the repressor and the inducers, they are functioning. So the first type of regulation is with the help of repressors. We can collectively call it as regulating with the help of repressors and inducers, where inducers are the substrates. So inducible enzymes, they usually function in catabolic pathways, and their synthesis is induced by a substrate molecule, that is, which can be called as a biochemical signal, biochemical molecule inducing the enzymes for its own degradative production, that is catabolism. And repressible enzymes, they usually function in the anabolic pathway like tryptophan, where their synthesis is repressed by the amount of the substrate already present in the environment, means high level or low level, so on and so forth. Now the regulation of such operons, that is tryptophan and lac operons, these involves negative control of the genes, because these operons are switched on and off by the repressor. A repression system is always negatively controlling mechanism, means it is always trying to, the RNA polymerase is always in a motion of trying to enter into the structural genes, and there is a constant block. Once this block with the help of repressor, as you can see here in the figure here, this is a lac repressor, and it is a beautiful dimer molecule, 2 dimers. So totally it forms a tetramer, 1 2 3 1 2 and 1 2 molecules. This is the core. DNA binding domains are there, means actually there are domains which go and club and attach at the site of the DNA. They remain there always. Tautomeric domains are there, and they just cling on to the DNA as long as the substrate molecule is not present there. So these are switched on and off in the presence or absence of the substrate. So that it's called as a negative regulation. Now the second example of regulation of gene expression is when there are 2 different carbon sources. The simpler one is usually preferred, so the other is automatically regulated. The other enzyme is automatically switched, as we saw in the example of carbon source available in the form of glucose and available in the form of lactose. So what actually happens is that lactose is, the lac operon is shut off, and the glucose operon, or the enzymes needed for utilization of glucose, is on. And here again a regulation is there, means shutting the lactose off and putting the glucose on. E. coli as example, if you take, it will prefer to use glucose when present in the environment, and when glucose is less, we have seen how the cyclic AMP receptor protein, CRP protein, and the CAMP receptor protein, that which is regulating it, acts as an activator of transcription. And this cyclic AMP receptor protein, that is the CRP protein, is activated when it binds to cyclic AMP. This activated CRP protein then attaches to the promoter, and it increases the affinity of RNA polymerase, that is, it enhances the activity of RNA polymerase, and it accelerates the transcription. This is all regulation, you see. How does it decide? Means glucose itself is deciding its own utility. When glucose levels they increase, if it is more, then CRP protein, it detaches from the lag operon, transcription proceeds, and if lactose is present, the whole process is very slow, or almost it can be off. So CRP, it helps in regulating not only lag operon but other operons also. You have studied lag operon in your syllabus. That doesn't mean that CRP protein is functional only for glucose and lactose utilization. This cyclic AMP receptor protein or the CRP protein, it helps in regulating other many operons which encode enzymes for catabolic pathways, arabinose operon, etc., like that. Utilization of other carbon sources, it helps in. So then again there is a regulation. So regulation with CRP, this is the second type of regulation which you should know. So the next type of regulation, the CRP protein can be second, this can be third, or you can just give your own sequences. Regulation by activators. Now this activators and enhancer is one terminology. Enhancers are usually used in eukaryotic systems. I will come to that. Even bacteria, they have enhancers. But regulation by activators is just what we saw, that how lactose itself acts as a activator. The substrate itself is a activator. So if lactose is present, if glucose is very low, and if the lag operon needs to be regulated, lactose molecule itself is a inducer, or you can say activator. We call lactose as a inducer. Lactose itself can be an inducer. There can be other activator molecules also, like the cyclic AMP. The cyclic AMP, when it binds to the CRP, it activates the CRP protein. So regulation by activator, that is what the second type is. Use of CRP, CAMP, and CRP activated by CAMP. So if CAMP is not produced, then this preferential utilization cannot be made possible. So this is regulation by activators. Again, see here, the inactive CRP, because there is no cyclic AMP, and if there is no cyclic AMP, this CRP won't bind to the operator. If it doesn't bind to the promoter, RNA polymerase will not function, because it is only in the presence of active CRP, it is activated by an activator, and that activator molecule is cyclic AMP protein. So unless and until this doesn't bind to the regulatory gene, RNA polymerase is not likely to get enhanced. So that is what the regulation by activators is. So that is the second type of regulation of gene expression. In the presence or absence of activators, they create a marked effect on the regulation activity. Now the third type, as I said, this is exclusively found mostly in eukaryotes, but most of the bacteria also, they show this type of activity. The activator gene, it is a gene cluster which is far away from the actual regulatory site. So there is a distance. How does this function? If there is, you can just watch my hand motions here. Here if there is a regulatory gene, there is a structural gene, regulatory gene, structural gene in this flow. Now some sequences upstream, there is a activator gene or enhancer gene, and this gene, I am saying, will control this gene. How does this thing, how this distance is maintained? That distance is maintained by this DNA bending like this. There are DNA bending proteins which fix here, and they will bend and bring the gene sequence. You can see the figure here now. This is a regulatory gene. Downstream there will be structural genes here, and upstream there are certain activator genes which bind to enhancers, activator proteins, and they enhance the promoter for binding up of the RNA polymerase and accelerating the RNA synthesis, mRNA synthesis. So away from the actual promoter site or the regulatory gene site, there are certain enhancer genes. These enhancer genes, there is a distance, so there is a distant control of these genes, controlling from the distance. How does that do? It's very interesting. See that there are certain DNA binding proteins. As I say, for the time being, remember these are mostly observed in eukaryotes. So there is a DNA binding protein which binds this section, bends this section of the DNA, and thus brings these enhancers and activators near to the regulatory gene site. And then there are a group of mediator proteins which will bind to this enhancer gene as well as the regulatory gene. It will come, and at the middle there will be a binding. So as you can see, this violet colored group of molecules, they have bound to the enhancer genes as well as they are bound to the regulatory gene, and then this facilitates the binding of RNA polymerase. And once it binds, it triggers off the RNA polymerase will enter into the structural genes. That is, the actual enzyme synthesis will start. So these are totally different types of proteins which are called as the enhancer proteins. These proteins enhance the rate of reaction of mRNA synthesis. It is usually found in eukaryotes and also in some types of bacteria. So this is how the eukaryotic system and some bacteria, they regulate the mRNA synthesis with the presence or absence of enhancer proteins and enhancer genes. So this is enhancement or enhancer control of bacterial transcription in E. coli. Very recently this has come into picture that there are certain sigma factors, and these sigma factors, they are combining with RNA polymerase. Means the RNA polymerase, before it binds to the DNA, it combines with sigma factors. There are certain alternative sigma factors which bind in different conditions, and that is another type of control, sigma factor control. I am not discussing sigma factor control here, but that type of control needs an enhancer protein. As you can see in the A picture here, pair of phosphorylated dimers are there. There is a dimer protein which binds downstream, in upstream of the region. Here there is a promoter. This blue color is RNA polymerase with a sigma factor, and this sigma factor is mainly responsible for bringing this enhancers which are upstream, some distant upstream of the regulatory gene. It attaches to the sigma factor, and then as you can see, there is a loop formation. There is looping of the DNA between them. Here there is no need for DNA bending proteins because this is a free DNA in prokaryotes. Okay, the enhancer proteins here, the NTRC dimers which are there, these bind and these contact the RNA polymerase by looping the DNA between them. This mechanism of transcription thus is activated by these enhancer proteins, and this process is similar to that of eukaryotes. To make you understand this enhancer control of bacterial transcription in bacteria or E. coli, I showed you the example which is similar to that of eukaryotes in the earlier slide. So this is the third type of regulation of gene control by enhancers. Enhancers are protein molecules, and remember, the enhancers, they will always bound to a DNA which is away from the gene cluster of the operon, that is away from the regulatory gene. The fourth type of regulation is by attenuation. This fourth type of attenuation, that is the one which we have seen in the tryptophan operon in E. coli. Transcription of the leader region of tryptophan operon, it occurs when the operon is repressed, we know, and this happens when there is tryptophan in the media, when tryptophan is present in the media. In the absence of the tryptophan, we have seen that transcription is not terminated in the leader region, and it proceeds through the entire operon. If tryptophan is absent, then the operon is on. And this leader region, which is just before the structural genes, is very important because it forms two different conformations, that is the hairpin or the stem and loop structure, which we called it as attenuators. For more detailed explanation of this, I request you to see the tryptophan operon. Whatever attenuation process is there, attenuation is formation of stem and loop structures in the operon model. That is one type of transcriptional control. In the presence of tryptophan, as you know very well, the hairpin structures, they are formed, then they act as terminators of the RNA polymerase. At another level, they also act as terminators of the movement of RNA polymerase also, as well as the ribosomes also. So we'll see the translational control in next lecture. But here the attenuators which are formed in the tryptophan operons, they are very good examples of regulation by attenuation, and regulation by attenuation is what functions at the transcriptional level. Because in the absence of tryptophan, different types of hairpin and loop structures, they are acting as anti-terminators for the RNA polymerase to function. It just stops and it breaks off even before entering the structural genes, as we have seen. This is the gene sequence, that is the leader sequence, and this is the leader mRNA, and it has a capacity of forming these types of attenuators which we have seen, the 1 and 2, 2 and 3, 3 and 4, and this does not allow, means if 3 and 4 is formed, automatically the RNA polymerase stops. That is regulation. And when does this thing happen? When there is presence of tryptophan in the medium. If tryptophan is present, there is no need for extra synthesis. To avoid energy wastage, to avoid wasteful synthesis, this has to be stopped. And this stops, as you can see here, 3 and 4 if it forms, then transcription terminates, and 2 and 3 if it forms, transcription proceeds. These stem and loop structures, 2 and 3, the stem and the loop structures. Loop structures are formed because there is no complementation in that, and the stem is formed because there are complementary regions in between the same strand of the mRNA. Actually, on the top, what you see as a single straight strand, this is folding in these fashions, and these foldings which forms the termination sequences are called as attenuators, and these are great structures which are involved in the regulation of gene expression. Means it decides whether transcription will continue or not continue, and this is basically what regulation is all about. So tryptophan operon is regulated by attenuation, you know very well. So the regulation of bacterial operon is usually taking place in a coupled transcription and translation process. So as you can see here, termination hairpin is formed, and this immediately relieves the RNA polymerase. So RNA polymerase will be deciding to enter into the structural genes or not on the basis of this terminator sequences of the hairpin and loop structures. So termination can be controlled via changes in RNA secondary structures, and this is determining whether RNA polymerase will enter into the production genes or not. So that is regulation. The same thing shown here again with two different terminator sequences, that is the 2, 3. There are many different terminator regions, different types of secondary structures in different types of bacteria. So that is an interesting point to be studied differently. So this is how the suppression of termination of mRNA synthesis takes place, and the regulation at the level of transcription is taking place. So you can see this gives a very clear picture here. So this mRNA, forget this for the time being, forget this translation by ribosomes. I had to show this in the figure here because in bacteria it is a coupled system, so it will move on here. One thing I would like to tell you, if near this laser marker, if there is a hairpin and loop formation, then termination of translation takes place. But today's lecture is termination of transcription. So if near the RNA polymerase which is acting on the DNA, near the RNA polymerase, if a hairpin and loop structure is formed, immediately termination of transcription takes place, and this is the regulation of transcription by attenuators. You can call it as hairpin and loop structure, you can call it as stem and loop structures, you can call it as simply hairpin structures, because in some bacteria the loop is not formed. If there is a complete complementation, there is no need for a loop formation. Depends upon what type of complementary sequences are present. So that is the fourth type. And the principal mechanism of attenuators, you know very well, is what I just said, the capacity to complement internally and form hairpin and loop structures. And these secondary structures, they are having the capacity to terminate the transcription. So you can just pause this slide and have a look at the various conditions of availability of tryptophan, which we have already discussed in the tryptophan operon. But you can pause the slide here and just look at the beautiful structures which the single length of mRNA internally folds to give rise to such structures. Now the next type of regulation is regulation by sigma factors. Now sigma is a element, a protein which binds to the RNA polymerase, sigma factor. It binds to the RNA polymerase and it decides whether the RNA polymerase will be active or inactive. Okay, so as you can see here, RNA polymerase, when it is bound with sigma, that is, it engages the RNA polymerase to bind to the promoter site. There is initiation, there is open promoter complex formation, there is elongation, and once these terminator regions are reached, the sigma factor, it is released from the RNA polymerase. This happens with all the operons. Try to understand. There are many operons, and this sigma factor is such an important protein molecule which, for a cell, there is a common RNA polymerase, but there are different types of sigma factors. RNA polymerase structure is same, but let us say there is a need for gene expression at high temperature. There is a different sigma factor activating this. If there is a need for a RNA polymerase to act in low temperatures, then there is a different type of sigma factor for that. If there is a gene expression needed in extreme conditions, how does the cell carry out that? There is a third type of sigma. There are different types of sigma factors which activate the same RNA polymerase in different types of environmental conditions. This is a very interesting aspect, and this is how regulation takes place. In different environmental conditions, in different types of situations, the protein synthesis is put on and off with the help of different types of sigma factors. So regulation by sigma factors, regulation of gene expression by sigma factor is one of the most important efficient activity by bacteria, which allows it to produce the gene in varied types of environments, extreme environments, psychrophiles, thermophiles, low nutrient conditions, high pressure conditions. Think about it. As many as environments are there, that many type of bacterial cells are living. But what about the protein synthesis in such conditions? Highly acidic conditions, highly alkaline conditions. How does the protein synthesis takes place? That is regulated. Regulation means in high acidic condition, protein synthesis has to be initiated. There is a different sigma factor for that which attaches and it initiates. In normal conditions, if it is let us say a normal neutrophile, and a neutrophile cannot grow in acidophile. Why? Because acidophile is the bacteria which has a special sigma factor which will attach, which will make its simple RNA polymerase special by allowing it to carry out the protein synthesis in highly acidic environment. So that is the speciality of sigma factor. So once the sigma factor, this is a general scheme here. In the next slide I will show you special sigma factors. So this sigma factor, when it attaches, then and then only this RNA polymerase is initiated and it synthesizes the mRNA, and the terminator sequences, when they are reached, instantly sigma factor is removed, and this sigma factor can reactivate this RNA polymerase for a second round of mRNA synthesis. Depends upon how much enzymes are needed. So that is one more interesting aspect of regulation by sigma factors. Now you can see here in this slide, if you just take example of E. coli has 7 different types of sigma factors which are needed for RNA polymerase for recognition, for the RNA polymerase to recognize the promoter and initiate transcription. 7 different types of sigma factors in one E. coli gene itself. Okay, if you recall the lac operon system, I had not mentioned earlier, but here I would like to mention lac operon. The RNA polymerase is initiated by a sigma factor called as S70. S70, which is the most commonly used sigma factor in E. coli. Now you can see there are a sigma 70, S54, S38, S32, or sigma 28, sigma 24, sigma 19, different types of sigma which are products of different genes. There are 7 different genes producing these 7 different sigma factors. And what is the role of this sigma factor? To go and bind at the site of promoter. Even before binding there, activate the RNA polymerase, which will go and bind at the site of promoter, and then it will be activated in that specific condition. Which condition? For example, sigma 70. Here transcription is initiated during exponential phase, during the log phase, when there is a rapid need for permease, beta galactosidase, like enzymes. These are just examples. What happens in the exponential phase, log phase? Huge substrate is coming, it is in a full phase of growth, and large amounts of enzymes are needed. So sigma 70 activates the RNA polymerase. Sigma 54, nitrogen regulated gene, NIF genes. Sigma 38, during starvation and stationary phase. Suddenly what happens? Sigma 70 is in log phase, then suddenly the substrate it gets depleted, then that sigma is removed, and then sigma 38 comes and binds. 38, what will it do? It will allow a slow and steady transcription of the genes needed during starvation or the stationary phase. Like that, heat shock, flagella, chemotaxis, response to the extracytoplasmic extreme heat, thermophiles, regulation of the ion chelating genes. So these you can see sigma factor is the one which controls the transcription by choosing the RNA polymerase. A very interesting aspect. So for the stationary phase there is a different sigma factor. For envelope capsule there is different. Ion metabolism there is a different sigma factor. Growth phase, flagellar regulation, chemotaxis, aerotaxis, whatever it is, phytotax, light tactic movements, nitrogen assimilation, heat shock, all these different conditions which the cell encounters, there are different types of sigma factors. So this use of sigma factor, the choice of sigma factor by E. coli is regulating the transcription process, mRNA synthesis. Okay, so that is what I showed in the earlier slide. The regulation, the sigma factor, the S70, sigma 70 usually combines, and the polymerase, prior to the RNA polymerase binding to the DNA, the S70, or there is alternative sigma factor called as sigma 54, which is in a different situation, binds to the RNA polymerase, and then the upstream genes, it has an enhancer and it enhances the production of mRNA. That is, the regulation of transcription takes place. So you can see this is how the regulation takes place, and there are various ways to regulate the transcription. Alternative sigma factors are there. These control the selective transcription of entire sets of genes. Means either these three are exclusively S70, S32 and S60. These are the various types of sigma factors in bacteria, all the types of bacteria. Earlier example was exclusively for Escherichia coli. Now this is general bacterial sigma factors. These sigma factors are called as transcription factors because they regulate the transcription. There are alternate sigma factors for different types of environment. Different sigma factors, they bind to RNA polymerase and they recognize a promoter region, that is, which is -10 to -35 base sequences away from the promoter, and it helps the DNA to bend and denature. That is, denaturation is called as melting. It helps the denaturation of the DNA. So open promoter complex formation is initiated by the sigma factors, and most of the bacteria, they have alternate sigma factors which function in different types of environment. E. coli has 17 and Bacillus subtilis it has 18 sigma factors. So in 18 ways it can regulate the transcription. In 18 ways it can control the transcription, that is the production of mRNA. Generally bacteria, they live in more dynamic environment, varied environments, and those such type of bacteria, they have many more types of sigma factors also. So we come to the end of this lecture where we have been discussing the regulation of gene expression at the transcription level. So just a recap for you to understand: regulation of gene expression, that is protein synthesis, at the level of transcription takes place at 5 different levels. There can be many more. These are not the only 5. There can be many more types of regulatory mechanisms. But for you to introduce you to this particular concept at the level of graduation, I have chosen these 5. You can explore even more. When you come to PG, when you will do research, you will be finding a much more open window here. A whole vista of knowledge will be thrown open at you in the form of research papers. And when you keep exploring research papers, you will see that these are not the only whole and soul 5 mechanisms. There can be many more mechanisms when it comes to the eukaryotic and prokaryotic world of protein synthesis. But just for the recap of today's lecture: regulation by repressors and inducers, regulation by activators, regulation by enhancers, regulation by attenuator sequences, that is the hairpin and loop structures, and regulation by sigma factors. So this is an interesting way of controlling the protein synthesis at the level of mRNA synthesis, that is the transcription. So I hope you understood these concepts very well. You always have an option to rewind, revise, re-listen a couple of times, and still if some doubts exist, you are open to ask me questions. I will be answering it however, whenever, wherever it is possible. I request you to go through the links which I have given below for some beautiful, much more knowledge of regulation, and also for preparation of notes. In my next lecture, I will be dealing with the regulation of gene expression at translational level. So till then, as I say, try hard to understand this beautiful mechanism in a small bacteria, a primitive bacteria, a single cell bacteria, which decides when a protein is to be synthesized and when not to be synthesized. Not only that much, what quantity is to be produced. So this is a beautiful concept which I hope you enjoy as much as I did by telling the concepts to you. So till the time we enter into the next lecture of regulation at translation level, I wish you all the best and happy understanding. Thank you.